Hello everyone, and welcome to today's live webinar, Cell Line Engineering with CRISPR-Cas9, Tips and Tricks to Maximize Success, presented by Laura Carleton, Scientific Liaison and Trainer at Horizon Discovery. And I'm Daniela Steele of Horizon Discovery, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Horizon Discovery. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like at any time during the presentation. To do so, simply type them into the Ask a Question box located on the far left of your screen and click on the Send button. We'll save several questions for Laura to answer during the Q&A session. Additionally, we have scientists standing by to answer questions as they come in during the presentation. We will answer as many questions as we can. If there are questions that we don't get to, we'll send an email response in the coming days. Additionally, when this presentation is available for on-demand viewing, questions can still be submitted and will be routed to Laura and the research team for an email response. Also, please notice you will be viewing the presentation in the slide window. To enlarge the window, click on the two-arrow symbol at the top right-hand corner of the slide. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem through the Ask a Question box. Continuing education credits are offered for attending this webinar. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. Also, check out the other tabs that contain helpful resources that may answer questions that will come up during this webinar. I would now like to introduce our presenter, Laura Carleton, who will be taking us through the tips and tricks learned through her years of hands-on gene editing experience. So, Laura Carleton is currently a scientific liaison and trainer at Horizon Discovery. Laura has extensive experience generating cell lines and model systems both in academia and industry, as well as a wealth of knowledge from working within a number of different disciplines, including recombinant protein expression, oncology, inflammation and cell death, and apoptosis research. Laura gained her PhD from the National University of Ireland in Galway, where she studied the pro-survival factor nerve growth factor, or NGF. In particular, Laura studied the effect of single point mutations on the affinity of NGF for its two receptors. Laura then went on to do her postdoctoral work in Lisbon, Portugal, at the lab of Colin Adrian, looking at intracellular trafficking of key inflammatory mediators to the plasma membrane. Since then, Laura has been working with Horizon as part of the gene engineering team, continuing her interest in using gene engineering and gene modulation techniques to generate optimal model systems. Laura has worked with many different cell types, as well as many different gene engineering goals, including knockouts and knock-ins of point mutations and tags. All of this together with her research knowledge means that Laura has a lot of hands-on experience in getting to a gene editing goal fast. So now for the presentation, and over to Laura.
Thank you for that kind introduction, Danny. So I'm just going to dive straight into the learning objectives of this webinar and what I aim to cover. So we are going to run through important considerations to make when initiating your gene engineering projects. And these will include the cell line optimization, considerations to take about your gene target, guide choice, position, and donor design. And finally, we're going to talk through different screening strategies and validation techniques that will help you find the important resultant clones. What I will be highlighting throughout this talk are the different tools and reagents that we actually use within the cell line engineering department here in Horizon to speed up this process and to kind of help us rapidly determine the genotype of our cell line. And this is how I'm going to also cover how we provide confidence in the knockout results with different secondary models and rescue models. So first we're going to go into broad detail on the possible applications of gene-engineered cells. So gene engineering is important for any researcher interested in determining the mechanism of action of their gene. So using gene engineering, we can make a gene non-functional, allowing us to look at that mechanism of action. Making a non-functional gene means that we can get a better understanding of how that gene is contributing to its specific biochemical pathway. And this is shown nicely here in this paper, where a gene was knocked out and its function was confirmed with a rescue experiment, so overexpressing that gene again. In addition to gene function studies, gene engineering can also be used to analyze protein-protein interactions. Gene engineering is essential to look at endogenous interactions in your cells, as it allows you to tag proteins and to visualize them or immunoprecipitate them at their endogenous level. And this is really crucial when looking at protein-protein interactions. Gene engineering can also be used as a validation technique where you can check the reliability of your key research findings that are obtained maybe with antibodies or RNAi. In addition to this, we can generate research tools using gene engineering, such as gene tags, as illustrated by this article by Daniel Lacker, where he showed how CRISPR can be used to tag genes in a really simplified and scalable model. And really, it's just gene engineering is just around in many different functions, and it's really had an explosion lately. However, gene engineering has been around for many years, and this is with zinc fingers, talons, and recombinant adeno-associated viruses. But there has been this explosion in gene engineering with the adaptation of CRISPR and CRISPR technology. So CRISPR has really made gene engineering less costly and less time consuming. So there really is no longer an excuse to work with a bad model. And that's why we've seen this explosion of gene engineering research. But what is CRISPR-Cas9? So CRISPR-Cas9 was originally described in bacteria as part of the bacterial immune system, and has evolved. The bacterial immune system has evolved this mechanism to defend against genetic parasites. So CRISPR essentially stands for clustered, regular, interspaced, short palindromic repeats, and these are areas within the bacterial genome. And Cas stands for CRISPR-associated proteins. So these are proteins associated with these clustered, regular, interspaced, short palindromic repeats. So this is a bacterial immune defense, and to fight off invading parasites, the bacteria are able to incorporate foreign DNA from the parasite into specific locuses in their genome, in these CRISPR locuses in their genome. The bacteria then express the integrated foreign DNA in these CRISPR locuses as guide CRISPR RNAs. These then interact with the Cas9 protein, and then the Cas proteins, along with the CRISPR RNAs, can make a complex that can bind to and cut the invading bacterial or virulent pathogen DNA. And it's this ability for the CRISPR proteins, for the CRISPR RNAs and the Cas proteins to bind to one another and to cut DNA in a double-stranded fashion that is the foundation for CRISPR gene engineering. So what essentially it is using these Cas proteins and these CRISPR RNAs to cut DNA. So CRISPR-Cas9 was adapted for the use in mammalian cells by essentially introducing this bacterial system into mammalian cells. Generally speaking, the adaptation of CRISPR-Cas9 has two forms. The first system that we see is on the left, and this has the Cas9 nuclease displayed in blue. This acts as a scissors to cut the DNA, and the Cas9 protein is complex with two small RNAs. The CRISPR RNA, often shortened to CRRNA or CRISPR RNA, and this is displayed in yellow, and the tracer RNA in purple. These small RNAs guide the Cas9 nuclease to a specific locus within the genome so the Cas9 can make the desired cut. The next system we'll talk about is this chimeric single guide RNA. So we can have the system on the right, which consists of the Cas9 nuclease protein and this chimeric single guide RNA or sgRNA, where the CRISPR RNA and the tracer RNA are linked together in a single molecule. And throughout this talk, I will be using guide RNA or gRNA as kind of an umbrella term throughout to cover both of these two products. So both Cas9 systems can be delivered or expressed in mammalian cells to cause DNA double-strand breaks. And it is these double-strand breaks which facilitate gene engineering. So for the gene displayed here, where the purple boxes represent the exons, so the protein coding regions of that gene, we have designed our guide RNA to bind within exon 3 of that gene. The Cas9 protein cuts the DNA and causes a double-strand DNA break. DNA double-strand breaks within the cell cause the activation of the DNA damage repair pathway. CRISPR gene engineering uses the cell's intrinsic DNA repair machinery to generate the genotypes and subsequent phenotypes that we desire. So for a knockout, when we want to make a non-functional gene, we generate a knockout using CRISPR gene engineering. So we allow these double-strand breaks that have been generated by the Cas9 and the guide RNA to be repaired by the endogenous repair machinery of the cell called non-homologous end joining. This repair machinery is quite error-prone and often results in small insertions and deletions within the cell. And it's these small insertions and deletions that can cause frame shifts in your protein coding sequence and result in a premature stop codon within your gene, making a non-functional protein. So it's really just how the cell repairs this Cas9 cut that generates these knockouts. To generate specific alterations in the gene, we generate what we call a knock-in using CRISPR. So we don't want the double-strand breaks in this instance to be repaired by non-homologous end joining, but by homology-directed repair pathway. And this is because it is a much more specific repair pathway where it uses a donor template to repair the DNA. So to increase the likelihood of the cells to repair their double-strand break by homology-directed repair, we co-transfect cells with a DNA repair template or a donor. This donor has high homology to our cut DNA, and as well as this, it contains the desired mutations, such as a single point mutation or a tag. The cell then uses this donor to repair its DNA template, and so it uses this donor to repair its DNA and to fix the DNA double-strand break. It also, because it's used this donor, it also allows for the incorporation of the desired mutations in this process. So because the donor contains these mutated regions, it incorporates these mutated regions into that fixed strand of DNA. And that's essentially how CRISPR works. It cuts DNA, and for a knockout to be generated, you allow that cut to be repaired by the endogenous cell machinery, but for a knock-in to be generated, you force the repair of that DNA using a donor template. So now that we understand the basic principles of CRISPR gene engineering, we are going to go through a typical cell line engineering experiment. So we understand how CRISPR works, but how can we actually get it to work in our cell lines? So if we look at the CRISPR gene engineering experiment in a cell line from beginning to end, there are a number of pitfalls in this project. And it's awareness of these pitfalls or risks within this experiment setup that will prepare you and to help you get that cell line faster. And it's really just understanding your cell line in the first instance and knowing your targeting reagents. So for knockouts, the gene engineering process usually takes 13 to 21 weeks, and for the knock-in, the project usually takes 18 to 26 weeks. The range is so large, and it can be actually much longer, because I've had a gene engineering experiment that has lasted over a year, and this is because it depends very much on the cell line that you will be using and the gene that you will be targeting. So one of the first crucial steps in a gene engineering experiment is preparation. So preparation includes your target reagent design, and this can take anywhere from 1 day to weeks and weeks, and this depends on your experience with CRISPR gene engineering, as well as how difficult your gene is to target. So you have to take into account a huge number of considerations before you can even begin to work with your cell line, and we will go through these in greater detail later, but this can be actually a real-time sync when you are performing your gene engineering experiments, and you could be sitting on your target reagent design for weeks and weeks. So after designing your perfect target reagents, you now need to initiate your targeting project. So if your cells are easy to transfect and select, this can take about 2 weeks. But if your cells have poor viability upon transfection of your targeting reagents, or if they have a very low transfection rate and therefore have a potentially very low number of edited cells in their percentage, then it can take much, much longer to grow and select up this population. So this process can take weeks and weeks and can be an unexpected time delay in your gene engineering experiment. So what we always do in the gene engineering department is characterize our cell lines to make sure that we can genetically engineer them and be prepared for these time delays and kind of hindrances to our research plan. So it's just all about the preparation in this area. So once you have your cell lines transfected and selected, you then need to single cell dilute them or perform a limiting dilution. And I've worked with a number of different cell lines, and some of them just really don't like to be single cell diluted. So you can have a very low recovery of your cells upon limiting dilution. And this can mean that you sometimes have to go back and design a completely new strategy for your cells after you've realized that they haven't single cell diluted. So it's really important to be prepared for this, and we always include this in our characterization procedure, which I'll go into in more detail a little bit later on. So if your cell line has single cell diluted and recovered and grown out, you can now expand these cells and check their genotype to screen for the phenotype of interest. And you can get a real mix of genotypes with CRISPR gene engineering, and this can make your screening quite difficult and a very time-consuming process. However, I will go through some of the tools to help you determine your genotype faster later on in this webinar. And finally, once you've got your 2 or 3 clones ready that have the genotype of interest and that are ready for expansion, we just need to expand and bank these cells. And this can take actually quite a long time, because you're going from quite a small population of cells, maybe in a 96-well plate or a 384-well plate, to a P175 or a triple layer flask. So this is actually a longer process than originally designed. But it's just about being aware of these possible time delays in your experiment. So there really is a large time variation in your gene engineering experiments. So it's about considering all these risks in your experiment, and these are what I'm going to go through with you today so that you can find your genotype faster and get your cell line faster. And the main considerations to take are the 7 displayed on this slide here. So we're going to go into each of these considerations, and what I'm going to highlight throughout this process are ways that we in the cell engineering team kind of minimize the time spent on each of these steps so that you can maximize your gene editing success. And these will be tools that Horizon have generated themselves in-house, as well as tools that we actually use from free online resources. So the first step there was to look at your cell line, so to characterize your cell line. So you want to consider, is your cell line engineerable? So questions you should ask yourself to determine if your cell line can be genetically engineered are, can I transfect my cell line, or can I infect my cell line? Can I enrich for the targeted events? Can they undergo limiting dilution to generate clonal populations? And is my cell line homogeneous? So for the transfection, I would recommend doing a small characterization procedure. And this will not only help for your cell engineering experiment, but for all further experiments with that cell line. We usually try both electroporation and lipid-based methods in the cell engineering department. And here I've just displayed an example of a cell line whose transfection efficiency ranged from 19% to 78% using different electroporation conditions. And usually once we have performed this transfection procedure, we use our FACS cell sorter to take out any of those GFP positive populations to see if our cells can be effectively sorted without any loss in cell viability. So what we have done here in this experiment in the bottom is we've transfected our cells with the fluorescent Cas9, and we're going to take that fluorescent population out and sort it into a multi-well plate to see if the cells are viable after sorting. And this will help us determine if we can get clonal populations straight from sorting, as well as if we can get clonal populations at all. However, if you don't have access to a cell sorter, you can do this exact same experiment, except you can transfect your cells using an antibiotic selection. The next question you have to ask about your cell line is, can my cells undergo limiting dilution? And should I allow my cells time to recover from selection and from kind of this rough cell sorting before I actually apply this additional stress to them? So we usually test a couple of different limiting dilution techniques in our cells. The image on the bottom is just an example of the recovery that I obtained when I seeded cells. This is a suspension cell line at 0.5 cells per well in standard media, so just the typical growth media that they use, as well as conditioned media, which is 50% media that the cells have been in for 24 hours and 50% their standard media. And what you can see and appreciate here is that I actually am getting better recovery and better cell outgrowth when I use the standard media. And this is just to highlight that the conditioned media isn't always the best way forward, and that sometimes doing this test can be extremely informative, because I have had cell lines not grow at all in their standard media or not grow at all in the conditioned media, and it really is cell line dependent. So it's really good to just do this test so you're not ending up with absolutely no clonal populations or outgrowth on your 96 well or 384 well plates. And finally, the last question I ask myself is my culture homogeneous? So in some cases, in some different cell lines, say, for example, in cardiomyocyte cells, you could have some cell lines, some cells within that culture that are actually contracting. In other cell lines, you could have semi-adherent cell lines, so you might be interested in maintaining that semi-adherent state or actually selecting a clonal population that has become 100% adherent. In another cell line population, you could have differentiated and undifferentiated cells. So it's just about knowing throughout your procedure that you're not selecting for one of these different phenotypes or genotypes, for one of these different phenotypes within your cell lines, because you want to be capturing the whole population and then finding out later on that because you've captured those two different kind of phenotypes in your cells, that has no effect on the gene that you're knocking out or you're modifying. So once you've characterized your cell line and found out that it can be genetically engineered, you should start thinking about your particular gene target, your particular gene that you're interested in. So one of the first things you should think about is, what is the effect of my modification on gene growth? Will it be lethal to the cell or decrease the cell viability? Will the modification change my phenotype? And if it will change my phenotype, can I actually select for that phenotype? And is it important to select the cells before single cell diluting them for this phenotype? The next thing you should consider is what is the copy number of my gene. So we all consider our genes to be a copy number of 2. That is that there's 2 alleles per gene, and we can see this here on the karyotype map on the right. And what a karyotype map basically is, it's an image that depicts all the chromosomes in the cell. And in this image on the right, you can see that we have 2 alleles per gene. So if we just look at the yellow genes just in the top left-hand corner, you can see that there's 2 genes for chromosome 1. There's 2 chromosomes, there's 2 chromosomes for chromosome 1. There's 2 alleles for chromosome 1, excuse me. And however, this is very rare genotype for your isolated cells in culture. So if you pay attention to chromosome 1 in this next insert, you can see in the next cell line, in this HeLa cell line, you actually don't have 2 alleles for chromosome 1, but you actually have 3. And you have a whole host of other genes that aren't reminiscent of any other chromosome down the bottom of the screen. And this is more of what we have in our general cell culture in any of these cell line adapted cultures that we grow as our model systems. So this is really important information to have at the start of your gene engineering experiment, because it can help you with your genotyping process. And how we usually detect the different copy numbers of our genes is we use droplet digital PCR or SNP6 analysis. So the next thing you want to consider is, now that you understand that your gene target and that you understand what your gene target is, that it won't increase the viability of your cells, and you know what copy number it is, you want to understand, has it got multiple transcripts? And what we usually do by testing to look at this is we actually do some bioinformatic analysis using Ensemble, and then this can be really easily found using Ensemble or gene cards. And what you want to be looking for is a transcript that is basically your CCDS verified, but you will understand what transcript you want to target. Usually it's the CCDS verified protein coding transcript. So the next question is, does my target have any known pseudogenes? And a pseudogene is an imperfect copy of a functional gene, and this can be kind of seen as incorporated anywhere within the genome, and it can really interfere with the genotyping and your targeting. So it is important to understand if your gene target has any known pseudogenes, and all of the known pseudogenes can be found in this link that I have here on pseudogene.org. And it's really useful information. It will help you guide your target reagent design later on. And the last thing that you should consider is, does my target have any difficult to amplify regions? So does it have any poly-Ts, high GC regions, or repeat regions? Because designing guide RNA into this region will be very difficult, as well as amplifying up this region if you want to do any kind of downstream genotyping. So I talked about the guide, but now we're just going to understand what makes a good guide and what really is the important considerations you should take when designing your guide. Essentially, a good guide is targeting the exon or intron that you're interested in. So we have different considerations to make when designing a guide RNA for a knockout or for a knock-in. So for a knockout, you want to have a guide RNA targeting the desired transcript, so that's one of the considerations we took earlier, as well as getting all the splice variants that are associated with that gene. You should also think about the possibility of targeting a functional region within that gene, and whether that transcript has any alternative start sites that could cause a truncated protein, even if you've edited upstream of that alternative start site. For knock-ins, the main consideration you want to take is to choose that guide RNA cut site as close as humanly possible to the engineering site. This will really increase the likelihood of your your modification getting incorporated into your gene. The general rule for all guide RNAs is to avoid DNA stretches that contain repetitive sequences, and you have to weigh up the on-target and off-target score. So you should really be aiming for a guide RNA that has a lowest possible off-target score and a highest possible on-target score. So there really is quite a lot of considerations to take when designing your guide RNA. So one of the other questions you need to consider is what algorithm to use. So there's a lot of online tools to design your guide RNAs, and we would really recommend that you use the guide RNA design algorithm that we have generated in-house. And this is a really powerful algorithm that takes into consideration all these key considerations, and it's constantly being updated, and it's really well maintained. And we have actually pre-designed CRISPR-RNAs and lentiviral sgRNAs using this algorithm. So these are different guide RNAs that you can just buy directly off the shelf for your gene of interest, and they're available for you. And I've just left a link to where you can get more details on those guide RNAs. But essentially, you can just put your gene of interest into the search bar, and that will bring up all of the guide RNAs that are available for your gene. And these are designed to have, these are guaranteed and designed to have editing. So they are guaranteed to have editing. So pre-designed guide RNAs are great for a knockout, but for a knock-in, the cleavage site has to be as close as humanly possible to that area, that alteration site. So you will probably need to design your own guide RNA for this. And as I said, we have this design algorithm available for you to use, and it is really user-friendly. So this is a free design tool that we have put on our website, and it helps you choose the most effective guide RNA for your specific genomic space. You can use this tool to indicate where you want to make that cut with CRISPR-Cas9, and then all the available guide RNAs will come up, and they will be ranked based on the functionality and specificity. I'm not going to go into too much more detail on this tool, but if you're interested in using it, I have included a link with more specific information and a really good how-to video about how to use this design tool. But it really is good. It's a well-curated, well-maintained algorithm, constantly being updated, and that's just good to keep in mind about using these free online tools. So I've run through the first 4 considerations: knowing your cell line, your gene target, guide position, and guide choice. And what I wanted to highlight to you here is that if you take advantage of Horizon's reagents, you can really decrease the time spent in designing and checking your reagents, and more time really using them, because we have already, with our reagents, these edited pre-designed libraries, as well as our guide RNA design tool, we have really kind of minimized the amount of time that is required for these tasks. So now that you have your guide RNA designed, the next consideration to take is the donor design. So for a knockout, you obviously don't need a donor design. However, it is a very good idea to think about at this stage your screening strategy that you're going to be using for your knockout. For your knock-in, you obviously have to consider a donor design, and there is a number of considerations to take. Mainly it is, as I'm going to say again, that your guide RNA cut site is as close as humanly possible to the knock-in location, so to the area that you want to modify. Another consideration is that to incorporate a screening strategy into your targeted population. So this could be a restriction site, or maybe a specific DNA region that you can amplify up later on. Or if your modification is going to have a different phenotype, you should be thinking about screening yourself based on that phenotype. So incorporating this, or thinking about this screening strategy at this stage, is going to be really well-deserved time, because it will really help you quickly identify the clones that you will be enriching for and screening for later on. And I'll have example of different screenings where it is very clear that a robust strategy was required for to make sure that we did get that cell line in the end. So another consideration to think of when designing your donor is to incorporate silent mutations into the guide RNA or the PAM site, and this will prevent the continued cutting of the Cas9 enzyme in your targeted population. So another question is the type of donor that you plan on using, and this can be plasmid or single-strand DNA oligos. And I have some more resources on this in the following 2 slides to help you choose between these 2. But in general, designing a donor can be quite an error-prone and time-consuming task, especially when you're first starting out in gene engineering. And I know very well the dread of ordering and buying the wrong reagents. So to help with this to streamline this process, Horizon has developed a tool to design both oligo and plasmid DNA donors. And to generate this tool, we've applied everything that we know about proper donor design and incorporated these rules into a very easy-to-use interface so that you can decrease the stress levels of designing your donor and decrease the errors that are often incorporated when designing these. So I'm just going to run through really quickly these oligo donor design and plasmid donor design tool. There are really good tools available for you to get more information on this, which I have linked here, but will also be at the end of the presentation in a resources slide. But I'm just going to give you an idea of what the oligo donor design can do for you. So all you have to do is to enter in your gene and your guide RNA sequence, define the type of alteration you're going to make, and then the tool will generate that proper oligo for you, even incorporating the silent mutations to prevent that ongoing cleavage of the Cas9. And so for your plasmid donor design tool, so we've split these up because they are different beasts. For the plasmid donor design tool, again, very similar setup. It just, as before, you enter your gene and your guide RNA information. You define along the gene where you would like to do your insertion, what kind of modifications you want to make, and the components required for the plasmid donor assembly are designed for you. And then you can just buy them. It's like one-click buy. So the plasmid donors are particularly good for knock-in of a fluorescent marker or other large inserts. And here are some other resources which we have generated in these links below to help you when designing your plasmid donor, and a how-to video on how to use that design tool. And again, these will all be summarized at the end of the presentation. This reduces the considerations that you have to make for your CRISPR design experiment even further. So we have kind of streamlined that donor design process. So it's really decreasing 7 considerations down to 3. And we generated these tools just to streamline this process and to make it quicker so that you're working with your cell lines quicker. So it really does help to use these tools and to basically take advantage of the experience that we have in CRISPR gene engineering to get your cell line faster. So unfortunately, we cannot do any of the screening for you. So you will have to determine your genotype faster using different screening strategies. But the best thing to do is to incorporate these into your into the design of the whole experiment. So there are a number of different ways which you can screen your cells. But at Horizon, we found that the best and most robust, quick and cost-effective way of screening is always PCR-based. So really looking at that DNA level. So for the knockouts, you can generate a mix of genotypes for your knockouts, and this can be very difficult to understand. However, there are a number of different online tools that we use to find the genotype of these knockouts in a really, really fast and cost-effective way. So in the cell engineering group, in general, if we suspect that the targeting can be is really low, and we would have been seeing this throughout the experiment, we usually perform a PCR over that edited area, so over that, say, exon 3 from the first example, and subject this PCR to T7 digestion. The T7 detects mismatched DNA and therefore can identify if that cell has been CRISPR edited. So it can identify if there has been some kind of insertion or deletion in that area. So we usually perform these on quite a large scale when we expect that there is a low low targeting efficiency in that cell. So if they've had a very low transfection rate, very poor selection, and if the gene is usually quite essential to the cell line, we usually perform these T7 assays. And then we take any of the positive clones forward, so any of the clones that showed T7 activity forward, and then we subject them to TIDE and CRISPR-ID analysis. So if we suspect a reasonable level of editing, we take 96 populations forward, perform a PCR over that gene-edited gene of interest, and we use these free online tools, TIDE and CRISPR-ID, to help us identify these edited populations. So this is just an example of CRISPR-ID, and essentially what CRISPR-ID does and TIDE do is that they deconvolve the mixed traces that we see during gene engineering and align them against the parental DNA to find out what is different in these mixed traces. So if you take the top parental DNA, you can see that there is the guide RNA site and the PAM site is just highlighted there for you. And then we look at the mutant clone that I have generated, and we can see here that there is a mixed trace in and around the guide RNA cutting site, suggesting that there has been an incorporation to that DNA, so that there's two different genotypes now in that gene. And then when we pass these sequencing traces through CRISPR-ID algorithm, what they do is they spit out the modification that is in the two alleles. So we can see here that on allele 1 we have a 6 base pair deletion, which is an in-frame deletion, and on allele 2 we have a 1 base pair insertion, which is an out-of-frame insertion. So we have one allele which is likely to be functional and one allele that is likely to have a premature stop codon. So it's really very quick way of finding out your real clones, your real knockout clones from your maybe heterozygous knockout clones or your homozygous in-frame deletion clones. It's really great tools. So for the knock-in, the experiments you should use for the genotyping strategy you should use is the one that you've incorporated or designed into your donor design step. And I'm just going to run through a couple of different knock-in strategies that I have used, and these are going to highlight the need for this robust screening strategy that we talked about earlier. So I'm just going to show you DNA gel electrophoresis of two different screens. So the first screen is here, So the first screen is here, and this is an example where the knock-in was smaller than the wild-type DNA. And I took advantage of this during my screening strategy, and I designed specific primers to amplify up both the wild-type DNA that is present, so this would be the high molecular weight, and the smaller low molecular weight band, which would signify any of the targeted cells that I had. So in this first screen, I screened 800 clones and I only got 30 populations which appeared to be positive for my target of interest. And in this second screen, where is it? There. In that second screen, I screened again 800 clones and I got 162 positive clones for my genotype of interest. And what is different in the second screen is that it is the same cell line, but it is actually a different guide RNA. So it really shows that there are differences between the different genes and the different guide RNA designs. So it's really best to take that guesswork out and use those off-the-shelf kind of guaranteed edited reagents. So in this knock-in screening strategy, I designed a primer so that it would only amplify up the targeted cells. So the primer only identified up when I had positive incorporation of my donor. So in this example, again, I screened 400 clones actually in this example, and I got 54 positive populations, which appeared to be positive for my targets. And in this example, I have 116 positive. And the difference between these two is that it is the exact same targeting reagents, however, it is different cell lines. And it really does show that the different cell lines, their different ability to even take up the CRISPR reagents and the targeting reagents can affect how they will perform later on. So now that we've screened our cells, we're going to go on to the validation. And this is, there is really no shortcut to the validation. You just have to do the validation. It's very time consuming, but it is very important to understand what you'd be working with. So to confirm the clonality of your cell, we, in the cell engineering, we use imaging software, which can be seen on the right here. So we can scan the cells, all the wells that we've done a limiting dilution in on day 0, and then look at them maybe 10 days after or whenever the cells have grown out to see if they're clonal or not. If you don't have this imaging software, you can also perform single-cell PCRs, or you can perform some topo cloning to make sure you're getting a good idea of what different alleles are present in your DNA mix, in your clonal population. So if there is a contaminant wild-type cells in your clonal population. So we usually confirm the genotype with a suite of different PCRs, as well as looking at the phenotype of interest. We like to identify any off-target incorporation as well if it is required. And this is just done by amplifying up those top off-target hits that we were notified of. We also confirm if there is no contamination of the bacterial cell, as well as the bacterial contamination, as well as the wild-type cell contamination, and confirm that there has been the suspected phenotypic changes within the cell. And during the validation procedure, we usually take quite a number of clones forward, and this is what is important because you want to be making sure that you're not drawing your results based on just one gene-edited event and one clone. You want to be having 2 or 3 clones that have been generated, that have this knockout or this knock-in, so you're not basically looking at just a clonal event of your cells. So to confirm the genotype of your, to confirm the phenotype of interest, we would recommend some Western blots validation. So on the left, to confirm the gene knockout of the selection of knockout cells, we used a suite of antibodies, and this was to confirm the presence of or absence of the protein of interest because these are knockout cells. And the Western blot data confirms the absence of the protein in the knockout cell line, and then this validates this gene engineering event. So you can see that on the knockout of the cells, which the cell line is basically represented on the top, you can see that on the knockout of that gene of interest, you do not get that protein band. But if you see a band on the Western blots, this does not actually confirm that the protein is functional, and this is where the gene editing has not completely prevented that protein from being expressed, but it might have generated a truncated or completely non-functional form of that protein. So it is really useful to perform additional studies that demonstrate that this protein that is functional, and you can do this by maybe looking at some immunofluorescence, so localization studies of where you expect that protein to be. So on the example of the right, you can see the presence of the absence of this knockout protein was detected using the immunofluorescence. In the wild-type cells, you can see the green staining, and in the knockout cells, you cannot see this green staining. And this would indicate that the cells are knockout for that gene SLC30A6. So now that you've generated your perfect research tool, we want to talk about the additional validation, so where you are validating your findings in a second. So you've generated your research tool, you've made your key findings, and now to kind of build more robust and confidence in your data, you can use the Horizon Knockout Catalog of cell lines as an additional model, as a secondary model for your gene engineering experiment for your hypothesis. So we have a huge amount of experience in in gene engineering, and this actually comes from the generation of these ready-made knockout cell lines as well as from custom products. But we have a knockout catalog of cell lines which allow you to increase the confidence in your results. And the catalog contains collections of knockouts for probing entire pathways, number of clones. There are a number of clones available per knockout gene, and the wild type control is also included free of charge so you can validate your results. I'm just going to go into a quick validation study that someone has performed using these cell lines, and it's just a really nice validation study where you can see that the difference between the wild type and the knockout was really clear, and they then rescued this knockout using overexpression, but also validated that a patient mutation was very important for the loss of function when they overexpress this protein again using the patient mutation. So this example involves the role of E3 ligases called RFWD3, and in E3 ubiquitin ligases are known to have a role in the DNA damage repair, particularly in homologous recombination. So it is critical for homologous recombination to repair defects in when they are subjected to a chemotherapeutic reagent. And here you can see that in the survival wild type cells, you can see that the survival of the cell was completely fine when they were subject to a chemotherapeutic drug. However, when we knock out RFWD3 in these cells, you can see that the survival of these cells was decreased massively. And then in contrast to that, you can see that once we overexpress this protein again, you have rescue of this phenotype, so the cells are staying resistant to this chemotherapeutic reagent. And what was validating this researchers' finding within the secondary model was that when you overexpress RFWD3 with these patient mutations, you do not get this survival anymore. So you do get this kind of cell death when applying this chemotherapeutic reagent. This is just the list of resources that I mentioned I would put at the end of the presentation. And if there are now any questions, I'd be happy to take them. But over to you, Danny.
That's great. Thank you very much, Laura, for this really informative presentation. As Laura mentioned, we'll now start the live Q&A portion of the webinar. And while we're getting that ready, we will be popping up a couple of questions on your screen. These really are very useful for so that we can plan for following webinars and understand what you would like to be seeing. So those questions actually I think will be coming up later on after the Q&A. But I'd like to remind you all that you can submit your questions by typing them into the Ask a Question box, which is located on the far left of your screen. Just type your question into the box and hit send. We'll try and answer as many of your questions as we have time for. So I see that a couple of coming in already. So should we start with the first question in the queue, which is, can you explain a little bit more about off-target effects? Should I be worried, and what can I do to minimize these effects? Laura.
Yeah, so to be on the safe side, we kind of have two belief systems that are important and simple ways to minimize potential off-target effects. So firstly, is to use the most up-to-date sgRNA design algorithms and selection of the guides with low predicted off-target potential. This is what I was talking about in the session about considerations to take when designing your guide RNA. So I really would recommend using guide RNA design algorithms that are constantly being updated and curated, and that would be obviously our design algorithm. But secondly, to kind of mitigate the possibility of effects of the off-target editing, one of the other things that we suggest people do is actually to use multiple clones or multiple different guides when looking at their knockout. So as I was saying, we would take several clones forward for validation of our several knockout clones forward for our validation, not just one, but maybe 4. And we would really test our hypothesis in those 4 cell lines. It's always what we'd recommend, or even ideally use 2 different guide RNAs for the knockouts and really test, multiple clones with multiple different guide RNAs, especially if it is a key finding. Or you could even use a secondary model, such as the HAP1 cell lines.
Great, thank you very much. I have another question, and it says, I want to create a secondary model to confirm my results. Can I just repeat the gene editing in a different cell line, or will I have to change any of the reagents or protocols? What do you think, Laura?
Yeah, so every cell line is different, and I think we went through that during the cell line characterization, but they can really vary on how easy they are to transfect or to single cell dilute. And you might have to screen many more clones for one cell line in comparison to the other cell line, which is one of the screening examples that I showed. But the best thing I could say is to really characterize your cell line, to make sure you're using the correct protocols for that cell line, but I would still use the same reagents, because you've already used and designed those reagents and you think they're great, and you've gotten a cell line using those reagents. But so you already that's already a known in your experiment. But if you want to then generate that same knockout in a different cell line, I would use those reagents, but use the protocols that are important for that cell line. So use the protocols that are going to make sure that you get that cell line in the end. So test their transfection, their single cell dilution, check if they can recover, and what's the best enrichment protocol to use for those cell lines.
Thanks, Laura. That's really helpful. It's difficult sometimes to find one thing that fits all, and I think you've been quite clear here how cell lines differ from each other. And I think it can be quite a common trap without looking into those individual protocols. Okay, one more question then. I'm interested in the ready-made knockout lines to confirm the findings from my own gene-edited cell line. How readily available are they? Thank you, Laura.
No worries. I'm glad you're interested in them. But we have over 3,000 ready-made knockout cell lines, and these are available for immediate delivery within like 7 days, depending on your shipping constraints in your country. But the catalog has a further 6,000 cell lines that can be manufactured on demand, and these are available within 12 weeks. So we just put these cell lines into our pipeline and generate them, and then they become available for everybody. So we're continually putting new cell lines, new knockouts as ready-made on available for our customers. So always request the cell line, and if the cell line that you're interested in isn't there, just ask our technical support. They're great. They just want you to get your cells as well, so they will help you find out the best cell line for you as well.
Thanks, Laura. And before we wrap up, I think we have time for one last question. It concerns validating the final edited cell line with antibodies. It reads, I've edited a frameshift mutation in my cell line and have confirmed that this has occurred with Sanger sequencing. How come I'm seeing a band in Western blot? I think you touched on that briefly in the presentation. I know, Laura, we've discussed this quite a lot, how unreliable antibodies can be. So this can be a tricky one. What do you think, Laura?
Yeah, we talk about antibodies all the time. Me and Danny talk about antibodies all the time. But there are a lot of non-specific nature. There's a non-specific nature to antibodies on the market. So it's really, really important to look at at the antibody that you're buying, see if it is actually validated in a knockout cell line, because there are a lot of antibodies out there that are actually validated in knockout cell lines or cell lines that have been modulated to have lower expression or higher expression. I would always recommend them. As well as that, if you do see a band on your Western blot, it doesn't necessarily mean that your protein is functional. It could be that the epitope to your antibody is designed upstream of the editing event, and therefore you're actually picking up the kind of the truncated version of your protein, however, and it was expressed. However, there is a completely non-functional region after that because it either has a stop codon or a completely different amino acid sequence. And it is the nature of Cas9 because you're actually just creating a cut in the gene. You're not completely deleting the gene. You're creating a cut in the gene to make a premature stop codon that you can get these kind of different results with your Western blot. But I'd always recommend a functional assay. And just to test a few different antibodies. Yeah.
That's really great, Laura. Thank you very much. So it's been a wonderful presentation and some great questions there. So thank you very much, Laura, and thank you to the audience as well for your participation. It's always the audience's questions that really make these webinars, I find. So it's a great job. Well done. I see that there are a few more questions remaining in the queue. If we didn't get to answer your question today, one of our scientists will respond to you via email. If you have further questions that you did not submit during the broadcast, then just email those to us at info@horizondiscovery.com. So that's Horizon Discovery, one word. Okay. But before we go, I'd like to thank the LabRoots team and Horizon Discovery for underwriting today's educational webcast. And this webcast can be viewed on demand all the way through December 2018. LabRoots will alert you via an email when it's ready to be replayed on demand when it's available. We encourage you to share this email with your colleagues who may also have missed today's live event. And as I mentioned, an exit poll will come up on your screen, and we'd really appreciate your feedback as it will help us plan for future webinars. So until next time, thank you very much and goodbye, everyone.
Bye, everyone.